good morning, everybody. I'm Laurie Burrows. I'm the Associate Director of the IIDR, and welcome to the 2025-26 version of our combined IIDR rounds. I'm very happy today to introduce our two speakers. So the first speaker will be Megan Tu, and Megan is a PhD candidate in Eric Brown's lab. And her research involves investigating the physiological trade-offs associated with antibiotic resistance. And by understanding how antibiotic resistance reshapes bacterial physiology, she's hoping to uncover novel vulnerabilities that can be used to treat resistant pathogens. So today she'll be talking to us about exploiting the fitness cost of carbapenem resistance. Our second speaker is Dr. Clayton Hall. He's the lead resident of the Medical Microbiology Residency Program at McMaster Hospital, or McMaster University. Sorry, not enough coffee yet, Clayton. He completed his MD and PhD at the University of Ottawa and his thesis on transcriptional regulation of antibiotic tolerance in my favorite organism, Pseudomonas aeruginosa, supported by a Vanier Canada grad scholarship. His current research interests include antimicrobial susceptibility testing, diagnostic stewardship, quality improvement, and patient safety. And today he'll be speaking to us about evaluating susceptibility testing methods for newer anti-pseudomonal antibiotics. So we're going to start with Megan and then switch to Clayton. And I would ask everybody, if you have questions, to please put them in the Q&A and we will answer them at the end of the two talks. So thank you. Megan, over to you.
Thank you for the introduction. So my project looks at exploring the fitness cost of carbapenem resistance. So I'm going to start by just giving a little bit of background information on carbapenem antibiotics, which are potent members of the beta-lactam family of antimicrobials and function by inhibiting bacterial cell wall synthesis. On this slide, I'm showing the structure of a carbapenem antibiotic, and here I've just highlighted the characteristic beta-lactam ring. Among beta-lactam antibiotics, carbapenems are of particular importance because they are considered to be the most reliable last resort treatment for bacterial infections. In other words, they're typically reserved for multi-drug resistant bacteria. However, due to the rise in multi-drug resistant bacteria, there has been a substantial increase in carbapenem usage over the past several decades, leading to the emergence of carbapenem resistance. In fact, the World Health Organization has declared carbapenem-resistant bacteria as the top priority for the development of new treatment therapies, highlighting the need to overcome carbapenem resistance. One of the primary ways that bacteria obtain resistance to carbapenems is through the acquisition of carbapenemases, which are enzymes that break down the backbone of carbapenem antibiotics. Broadly speaking, carbapenemases can be divided into two categories based on their mechanism of beta-lactam ring hydrolysis. The first category are metallo-beta-lactamases, and common examples include IMP, NDM, and VIM-type enzymes. These enzymes use zinc ions to activate a nucleophilic water molecule to hydrolyze the beta-lactam ring. The second category are serine beta-lactamases. An example includes OXA48, and these enzymes have an active site serine residue that covalently binds to the beta-lactam ring in order to open it up and confer resistance. However, expressing antibiotic resistance, although advantageous to bacteria in the presence of antibiotic pressure, can often be associated with a fitness cost. And simply put, this is just because expressing resistance genes is a very resource-intensive process that may have impacts on bacterial physiology. If we can identify, though, and understand the fitness cost associated with antibiotic resistance, in this case, carbapenem resistance, we might be able to develop new strategies to treat resistant pathogens. So there are a few different ways we can go about assessing the fitness cost of antibiotic resistance. The first would be to monitor growth in vitro. So you have a susceptible population, you're highlighted in orange, as well as a resistant population, highlighted in blue. And if resistance were to negatively impact bacterial physiology, you would expect that resistance would result in a growth defect relative to our susceptible population. The second strategy is to perform a competition experiment in vitro. So you start off with equal parts of your susceptible and resistant strains and measure the ratio of the two strains over time. If the susceptible population outcompetes the resistant population, this might indicate that resistance is associated with a fitness cost. Additionally, you can perform the exact same experiment in vivo. So you inoculate mice with equal parts of your susceptible and resistant strains, and at a given time point, you look at the ratio of both isolates. And if there's a fitness cost or an in vivo relevant fitness cost associated with the expression of resistance, you'd suspect that the susceptible population would outcompete the resistant one in establishing an infection. So in assessing the fitness cost of carbapenem resistance, we decided to leverage nutrient-limited conditions. And this is because nutrient-limited media, such as M9 minimal media, we believe is more reflective of the environment that bacteria encounter within the host population and might unveil unique or previously undiscovered fitness costs associated with the expression of antibiotic resistance that one might miss under standard nutrient-rich conditions, such as MHB. And with that, my talk is broken down into 3 different parts today. So the first is to assess the fitness cost of carbapenem resistance in vitro as well as in vivo, determine the mechanism of the fitness cost that can hopefully guide the development of novel therapies to treat carbapenem-resistant bacteria. So to start assessing the fitness cost of carbapenem resistance, we were able to leverage the antibiotic resistance platform. And for those of you who don't know, this is a wonderful platform developed by the Wright Lab. And it's this library of approximately 100 different antibiotic resistance genes that have been individually cloned into an identical genetic background. So we started by assessing the fitness cost of genes that confer resistance to beta-lactam antibiotics, focusing on those that confer resistance to carbapenems primarily. Before my talk today, I'll be focusing on the metallo-beta-lactamase VIM2 as it produced the most pronounced fitness cost in vitro. So we started off by assessing the growth of E. coli expressing VIM2, shown here in blue, and compare that to growth of E. coli carrying the empty vector, shown in red, in M9 minimal media. So again, this is a nutrient-limited environment. And we observed that expression of VIM2 led to a growth defect indicated by an increase in leg time, as well as a decrease in growth rate. As VIM2 metallo-beta-lactamase requires zinc to confer resistance, we had hypothesized that it was the lack of zinc in M9 minimal media that led to this phenotype. When we supplemented zinc back into the media, we were able to suppress the cost of VIM2 expression, leading to comparable growth between our control and our resistant strain. To understand how widespread this fitness cost was across Enterobacteriaceae species, we transformed the plasmid and the corresponding empty vector into clinical strains of E. coli, Salmonella, Klebsiella aerugines, and Klebsiella pneumoniae. And we monitored growth in M9 minimal media, comparing the leg time as well as the growth rate to the empty vector control. And we can see across all strains tested that expression of VIM2 led to an increase in leg time as well as a decrease in growth rate, similar to previously observed. Additionally, this growth defect was linked to the fact that M9 minimal media is a zinc-deprived environment, as adding zinc back into the media suppressed the growth defect, leading to more comparable leg time and growth rate to our empty vector. Moving forward, we decided to focus on the pathogen Klebsiella pneumoniae expressing VIM2, primarily because there are several examples in literature of this strain harboring VIM2 in clinical situations worldwide. And so to understand the physiological relevance of this fitness cost, we performed a competition experiment in human serum, a potentially zinc-deprived environment, between Klebsiella pneumonia harboring the empty vector and expressing VIM2. And we observed that the control strain, the empty vector strain, substantially outcompeted Kleb expressing VIM2, indicating a growth defect again. And similar to M9, the addition of zinc was able to suppress this growth defect, leading to more comparable colony-forming unit counts between our resistant and our control strain. Next, to evaluate the impact of VIM2 expression on pathogenesis, we used a murine infection model. So we performed a competition experiment where mice were co-infected with Kleb carrying the empty vector and Kleb expressing VIM2. And at a 7-hour time point, we looked at the ratio of both of these strains. And so here I'm showing the competitive index, which is just essentially the ratio between VIM2 expressing Kleb and Kleb carrying the empty vector. So a lower competitive index means that there's less VIM2 bacteria and therefore a higher fitness cost. It was really exciting to see that across every single site of infection that we looked at, VIM2 expression did lead to a decrease in pathogenesis relative to our control. A competitive index of 1 would indicate equal levels of growth. We wanted to understand whether or not this fitness cost was linked to the fact that the in vivo environment might also be zinc limited. So we also repeated this infection model, but with concurrent treatments of zinc sulfate. Indeed, treating with 20 mg per kg of zinc sulfate was able to suppress the fitness costs associated with VIM2 expression in vivo, leading to more comparable growth between our resistant and susceptible strain. And this effect appeared to be dose dependent, as treating with a higher concentration of zinc sulfate further enhanced the growth of VIM2 expressing CLAP. We do notice a spike in counts in our kidney and liver. We just hypothesize this is because zinc might accumulate at these sites in the body when treated at such a high dose. So now that we've assessed the fitness cost of VIM2 expression, both in vitro as well as in vivo, we were curious. We wanted to understand the mechanism of the cost now. So to understand the physiologic response of E. coli to VIM2 expression, we conducted RNA sequencing. So we identified 176 differentially regulated genes in VIM2 expressing bacteria, 83 of which were upregulated, 93 of which were downregulated. Of note, the genes involved in cell motility, the phage shock response, the heat shock response, as well as the CPR X-regulant were all differentially regulated. Blue in this case indicates downregulation, whereas red indicates upregulation. I know there's a lot going on in the slide, but all I want you to take away is all of these processes are linked to cell envelope stress. So this data, in combined with previous literature, suggests that VIM2 expression VIM2 under zinc limitation might be misfolding in the periplasmic space of gram-negative bacteria, leading to the growth defect that we previously observed. And so a hallmark of envelope stress is altered morphology. So we also took this opportunity to visualize what E. coli expressing VIM2 might look like relative to our empty vector control. After growth in M9 minimal media, E. coli harboring the empty vector appeared predictably rod-shaped, whereas E. coli expressing VIM2 had this filamentous phenotype, which we thought was really interesting. And similar to before, the addition of zinc was able to suppress this altered morphology, leading to rod-shaped bacteria in both E. coli expressing VIM2 as well as our empty vector control. What was really exciting to see is that in more clinically relevant bacteria, such as Klebsiella pneumoniae harboring VIM2, we also get this altered morphology. Excuse me. So with this in mind, we had hypothesized that VIM2 expressing bacteria might be hypersensitive to perturbations in cell envelope stress response pathways. However, to answer that question, we decided to take more of an unbiased genomic approach to identify potential targets for VIM2 expressing bacteria. To that end, we modified the plasmid that harbored VIM2, as well as a corresponding empty vector, to make it suitable for high throughput conjugation into diverse genetic backgrounds. Specifically, we wanted to move both of these plasmids into the KO collection, which is comprised of about 4,000 non-essential gene deletions, as well as the CRISPR-I collection, which harbors 350 essential gene knockdowns. After moving both of these plasmids into these genomic collections, we then transferred each collection onto M9 minimal media, and we are looking for gene perturbations that selectively reduce the growth of VIM2 expressing bacteria, therefore enhancing the fitness cost associated with this enzyme. We are able to identify 126 non-essential gene deletions and 43 essential gene knockdowns that enhance the growth defect. And so here's just a visualization of the data. Both of these are index plots. On the left is of the KO collection, on the right is of the CRISPR-I collection. And along the y-axis of both graphs is the normalized growth of the collection harboring VIM2 relative to the empty vector control. So each point represents a unique or an individual gene perturbation, and all points highlighted in red are gene perturbations that significantly enhance the growth defect associated with VIM2 expression. An analysis of all gene perturbations that enhance the fitness cost actually revealed a significant enrichment in genes required for cellular response to cell envelope stress, which aligned with our previous mechanistic data and was really great to see. Among these genes were specifically gene perturbations that suppress the activity of the sigma E envelope stress response pathway. So to give a little bit of an overview of this pathway, the sigma E envelope stress response pathway acts to prevent disturbances in periplasmic protein folding. Specifically, DAG-S will sense misfolded proteins in the periplasm, in this case, misfolded VIM2, leading to several downstream events that releases sigma E into the cytoplasm, where it can regulate gene transcription. Ultimately, this pathway culminates in the transcription of DAG-P, a periplasmic protease. We hypothesized that DAG-P is crucial for mitigating the fitness costs associated with VIM2 expression by degrading misfolded VIM2. However, to test the importance of this gene in VIM2 expressing bacteria, we conjugated both the empty vector control as well as VIM2 into a DAG-P deletion mutant and monitored growth in M9 minimal media. It appeared that deleting DAG-P actually was lethal in a VIM2 background, suggesting the importance or highlighting the importance of this pathway in VIM2 expressing E. coli. As VIM2 expressing E. coli was hypersensitive to deleting DAG-P, we hypothesized that VIM2 expressing bacteria would also be hypersensitive to compounds that inhibited this pathway. And so we were luckily able to get our hands on two compounds, the first of which targeted RSEP, and the compound was termed betimestat. Betimestat displayed at least an 8-fold increase in potency against VIM2 expressing bacteria, which is in line with what we had hypothesized. Additionally, we were able to find a DAG-S inhibitor, which displayed very similar activity, showing an 8-fold increase in potency against VIM2 expressing E. coli. It was exciting to see that the DAG-S inhibitor also maintained activity against Klebsiella pneumoniae expressing VIM2, allowing us to study this inhibitor in vivo. So to test the therapeutic angle of inhibiting the sigma E stress response pathway in vivo, we conducted a murine systemic infection model whereby mouse were either infected with Kleb harboring the empty vector or expressing VIM2 and subsequently treated with the DAG-S inhibitor. Although the DAG-S inhibitor did decrease the bacterial load of our empty vector strain, likely owing to the fact that DAG-S is an essential protein, it substantially decreased the bacterial load of VIM2 expressing Kleb, or showed preferential activity against this strain. We then wondered whether induction of envelope stress response pathways might actually sensitize VIM2 expressing bacteria to other antibiotics, particularly other FDA-approved antibiotics that might be repurposed. And so we screened a variety of antibiotics for increased potency against VIM2 expressing E. coli in M9 minimal media. So here's a subset of the antibiotics that we screened, spanning diverse chemical classes. And along the y-axis, we're showing the full change in MIC, or the minimum inhibitory concentration. In this case, the higher the full change, the more potent the antibiotic was against VIM2 expressing bacteria. Of particular interest were macrolide antibiotics, which target the bacterial ribosome. But interestingly, macrolides typically display low activity against gram-negative bacteria, such as E. coli, due to poor accumulation inside of the cell. However, these compounds showed enhanced potency against VIM2 expressing bacteria. Similar to previous results, the addition of zinc was able to suppress the activity of these antibiotics, indicating that antibiotic susceptibility in this case was linked to the fitness costs that we had previously observed. Among the macrolide antibiotics tested here, we decided to follow up on azithromycin due to its favorable safety profile in mice, as well as widespread use in the clinic. So like I mentioned before, azithromycin, as well as other macrolides, are typically prescribed for the treatment of gram-positive bacterial infections. And just to highlight the clinical relevance of this antibiotic, in 2020, it was estimated that there were over 10 million prescriptions of azithromycin in the United States. We then wanted to uncover why VIM2 expressing bacteria were more susceptible to azithromycin. Our first hypothesis was that potentially VIM2 expression might increase compound entry into the cell. To answer this, we performed an intracellular accumulation assay, revealing the fact that azithromycin did significantly increase more inside VIM2 expressing bacteria, likely leading to enhanced antibiotic susceptibility. However, we then wondered, how does azithromycin accumulate inside the cell? Potentially, VIM2 expression might disrupt the outer membrane, thereby letting more azithromycin enter the cell. And to characterize the impact of VIM2 on outer membrane integrity, we then measured the uptake of NPN, a hydrophobic fluorophore. So typically, an intact outer membrane prevents entry of NPN into the cell. However, if VIM2 expression were to perturb the outer membrane in any way, NPN would be able to enter the cell where it interacts with the phospholipid layer and subsequently fluoresce. So in this assay, higher fluorescence means more membrane perturbation. That's exactly what we had seen. So NPN fluorescence was increased in a VIM2 background, suggesting that VIM2 expression perturbs the outer membrane of gram-negative bacteria, letting more azithromycin enter the cell and therefore kill. Additionally, the subsequent supplementation of zinc into M9 minimal media suppressed outer membrane perturbation, aligning with previous antibiotic susceptibility data. So to evaluate the therapeutic potential of azithromycin in treating VIM2 expressing bacteria, we conducted another murine infection model. So at 0 hours, we infected mice with either club carrying the empty vector or club expressing VIM2, followed by subsequent treatments of azithromycin. Azithromycin had limited activity against our empty vector control, but significantly reduced the bacterial burden associated with VIM2 expressing, which was really exciting. The last question that we posed is whether or not sensitivity to azithromycin is maintained in clinical isolates expressing VIM2. So up until this point, we have used an artificial plasmid that constitutively expresses VIM2, but wanted to see if isolates that naturally express VIM2 also display the same phenotype. So we were able to get our hands on several clinical isolates that naturally express VIM2. I do want to mention, though, that these isolates are actually Pseudomonas isolates. Although club expressing VIM2 does exist in the clinic, it's relatively rare in North America, so just couldn't test any of those. Nonetheless, as these are all Pseudomonas isolates, we used PaO1 as a control, as this isolate or as this strain does not harbor VIM2. And we started off by characterizing azithromycin susceptibility, or azithromycin MIC, in M9 and M9 supplemented with zinc. We hypothesized that if these clinical isolates behave in line with our previous work, we would suspect azithromycin to be more potent under zinc-deprived environments. And that's exactly what we observed. But for majority of the isolates that we tested, there were a subset of isolates that did not display enhanced potency or enhanced susceptibility to azithromycin under zinc limitation. And to understand this variability in azithromycin susceptibility, we decided to quantify VIM2 expression levels in each isolate using qPCR. And we did observe that increase of sensitivity to azithromycin correlated with higher VIM2 expression, which was really cool to see. But then we wondered, is there a way that we can elevate VIM2 expression levels in these isolates, thereby increasing sensitivity to azithromycin? When we exposed isolates to a subinhibitory concentration of meropenem, a carbapenem antibiotic, we were able to elevate VIM2 expression levels, which subsequently sensitized these isolates to azithromycin. Lastly, we also wanted to evaluate the therapeutic potential of azithromycin to treat a clinical isolate that naturally harbors VIM2. So we had selected CDC 243 for further follow-up, as it displayed the most susceptibility to azithromycin under zinc-limited environments. So after infecting mice with CDC 243, we followed it with one treatment of 50 mg per kg of azithromycin or a vehicle. We were able to show that azithromycin was able to decrease the bacterial load of this isolate by 6 logs relative to vehicle treatment, effectively clearing the infection if 3 of the mice tested. Additionally, concurrent treatments with zinc was able to suppress the activity of azithromycin against this isolate, further validating our in vitro findings. So overall, we demonstrate that expression of VIM2 reduces bacterial fitness, both in vivo as well as other physiologically relevant environments. And this fitness cost actually sensitizes cells to compounds that are typically inactive against gram-negative bacteria, including azithromycin, again, both in vitro and in vivo, supporting potentially novel therapies to treat VIM2 expressing bacteria. And with that, I would like to thank my lab as well as everybody who has helped with this project. And I would like to give a special thanks to my supervisor, Dr. Eric Brown, for all of his guidance. Thank you.
Thanks, Megan. That was awesome. Over to Clayton.
Hi everyone. I'm just getting over a cold, so sorry if my voice falls out as I talk. Thank you so much for having me speak today. Today I thought I would talk about some work that I did at Shared Hospital Laboratory at Sunnybrook, where we evaluated phenotypic and genotypic susceptibility methods for newer anti-pseudomonal beta-lactamase inhibitors, and some of the challenges that we encountered with these methods. So I thought that I would start off with a case. So this was a patient who I've seen previously, severe peripheral vascular disease. They ended up having acute limb ischemia because of that, needed an axillofemoral bypass, had a prophylactic calf fasciotomy to prophylax against compartment syndrome. And then unfortunately, that fasciotomy wound became infected with Pseudomonas aeruginosa that was initially quite susceptible, but increasingly became more resistant, and that infection became chronic, ultimately became resistant to all first-line agents. And I'm just showing a screenshot of the susceptibility report, where you can see that all of the first-line agents that we would use against Pseudomonas are resistant. Because of that susceptibility report, this patient went on to have an above-knee amputation with a curative intent, but unfortunately, that stump of their above-knee amputation became recurrently infected with that same resistant Pseudomonas aeruginosa as had been in the fasciotomy wound. The isolate was sent to a reference laboratory for susceptibility testing of some newer anti-pseudomonal agents, and the results took 7 to 10 days to return. So this is not an uncommon clinical scenario for us, increasingly, where we're seeing this MDR Pseudomonas aeruginosa, and we're having to send to a reference laboratory for additional susceptibility testing, which does have quite a lengthy turnaround time. Just an overview of beta-lactam resistance and Pseudomonas aeruginosa. The vast majority of beta-lactam resistance, particularly in the North American setting, is due to adaptive evolution of a large repertoire of chromosomal resistance determinants. So I'll highlight here for you AMP-C overexpression. So Pseudomonas has an intrinsic AMP-C enzyme called Pseudomonas-derived cephalosporinase that can become upregulated, and this confers resistance to things like piptazo, ceftazidime. MexAB OPRM is an important efflux pump in Pseudomonas aeruginosa, and again, mutational upregulation of this efflux pump can confer resistance to a number of antibiotics, including meropenem carbapenemase. And then the last one that I wanted to highlight here for you is OPRD, which is a porin in the outer membrane of Pseudomonas that is important for imipenem uptake into the periplasmic space. So OPRD loss of function does confer imipenem resistance in clinical isolates. Fortunately, carbapenemases as a cause of resistance in Pseudomonas remain rare in the Canadian setting. More recent data suggests less than 5% of carbapenem-resistant Pseudomonas harbor a carbapenemase. That being said, the story is very different in many other parts of the world. Spain, for instance, 30 to 50% of their carbapenem-resistant Pseudomonas harbors carbapenemases, in particular your class B metallobeta-lactamases. Fortunately, newer anti-pseudomonal beta-lactam inhibitors, which we'll abbreviate as BLBLIs, have been developed to help address some of the more common resistance mechanisms, in particular PDC upregulation that we see in clinical isolates. And the newer agents take on two flavors. So the first flavor is you can have a new beta-lactam combined with an old beta-lactamase inhibitor. So the best example of that is ceftolozane tazobactam, which is abbreviated CT. The trade name is Zerbaxa. Ceftolozane is a modification of ceftazidime, and I'm just showing the two molecules below here. And you can see this bulkier side chain that I've highlighted in ceftolozane that was specifically designed for steric hindrance of PDCs in Pseudomonas. Tazobactam in this combination has a limited role as the beta-lactamase inhibitor in Pseudomonas, but it is important for other organisms, including Enterobacterales. And this agent has been Health Canada approved since 2015, and we do make use of it clinically in these scenarios of pan resistance to first-line agents. The second flavor of BLBLIs that have been developed are combining an old beta-lactam with a new beta-lactamase inhibitor. The first example of that was ceftazidime avibactam, abbreviated CZA. The trade name is Avicaz. Avibactam is a first-in-class diaza bicyclooctane beta-lactamase inhibitor, and unlike prior beta-lactamase inhibitors, it's not a beta-lactam itself. It inhibits PDCs, it can inhibit ESBLs, and it can inhibit class A, C, and D carbapenemases. It has no class B activity. And it rescues ceftazidime when PDCs are overexpressed. So when PDC is overexpressed, it chews up all the ceftazidime. in combination with avibactam, you're inhibiting those PDCs even though they're overexpressed, and you're rescuing ceftazidime activity. This is available through Special Access Program, but we have been able to obtain it clinically. And then the next example is imipenem relebactam, abbreviated IMR. The trade name is Recabrio. Relebactam is a newer diazobicyclooctane beta-lactamase inhibitor. It's sort of the next generation of avibactam. It also inhibits PDCs, ESBLs, and class A and C carbophenamases. It has limited class D activity and again has no metallo-beta-lactamase inhibitory activity. And this one works through a little bit of a paradoxical mechanism because I just told you in the slide prior that imipenem resistance is predominantly OPRD mediated, not beta-lactamase mediated. That being said, in the setting of OPRD loss of function plus PDC overexpression, the overexpressed PDC can be a little bit overambitious in its profile, and it can have low-level imipenem hydrolase activity. And in the setting of low imipenem concentrations in the periplasm because of OPRD loss of function, the PDC overexpression actually does become important for imipenem resistance in that setting, and that's why relebactam is useful to rescue imipenem susceptibility. And this, again, is available through the Special Access Program. Previously, resistance to these agents was quite rare, but we do know that resistance is increasing, and I'll show you some semi-local data showing that shortly. Because of that, many labs did not perform in-house testing of newer beta-lactam, beta-lactamase inhibitors, and they were referred out to reference laboratories. So for instance, in our setting, we would refer out to public health, where they perform broth microdilution method for susceptibility testing. And this can take about 7 to 10 days to come back. Resistance, like I said, is well described to all agents globally. Canadian data is lacking. But the risk here is that with the 7 to 10-day turnaround time, patients either have already completed an empiric therapy and they're better, or they may have decompensated on an inactive regimen before we have AST results available. So I did an elective at Shared Hospital Laboratory earlier this year. It's one of the largest regional microbiology laboratories in Ontario. And their first question was, what is our resistance rates to all of these newer beta-lactam, beta-lactamase inhibitors? So we had 183 consecutive MDR Pseudomonas isolates from 2023 to 2024 that had all been referred out for CZA, CT, and IMR testing by public health by broth microdilution. And at Shared Hospital Laboratories, MDR Pseudomonas is locally defined as resistance to all of ciprofloxacin, ceftazidime, and piptazo, which are important for first-line agents. Here I'm showing the MIC distribution of isolates by broth microdilution at Public Health Ontario. The bars colored green are susceptible. In yellow, you have intermediate, and in red, you have resistant. In the first graph up here on the upper right-hand corner, I'm just showing the ceftolozane tazobactam MIC distribution. And you can see that we had 7.1% of those 183 isolates that were ceftolozane resistant. In the lower left-hand corner here, we're seeing ceftazidime avibactam, and there we saw 31.7% of isolates were actually ceftaz-avi resistant, and in those patients, there was no pre-existing ceftazidime avibactam exposure. And then finally, in the lower right-hand corner here, we have imipenem relebactam. I'm showing in the solid bars imipenem relebactam, and in the hatched fill, I'm showing imipenem, just so that you can see how relebactam nicely shifts the MIC profile of imipenem to the left. And there we saw about 10.4% resistance in those 183 isolates. So overall, ceftolozane tazobactam was the most active agent in this isolate collection. Cross-resistance was quite common given the shared mechanisms of resistance to these agents, which we'll talk about a little bit later. But among the isolates that had cross-resistance, ceftolozane tazobactam was still the most active. Given that we saw quite a sizable proportion of MDR Pseudomonas aeruginosa isolates that were not susceptible to beta-lactam, beta-lactamase inhibitors, and that these are the isolates that we want to have susceptibility testing for because they're the isolates that will be treated with these agents clinically, we felt that a turnaround time of up to a week was too long for patient safety, and that you really could not choose an empiric therapy confidently. So because of that, we next asked, could we do the susceptibility testing in-house for MDR Pseudomonas aeruginosa? So we took two approaches. The first was, could we do phenotypic testing using a commercial system called the Phoenix M50, which is by Becton Dixon, that was already available at shared hospital laboratories and is a system that is used for routine susceptibility testing there. And then we also did an exploratory analysis, given the interest of shared hospital laboratories for whole genome sequencing, to see if we could develop a genotypic testing method using AMR Finder Plus analysis to predict susceptibility or resistance to all three of these agents. There are a few things, definitions to be aware of whenever we're comparing susceptibility testing systems to, in this case, reference broth microdilution, which was performed at Public Health Ontario. So the first is categorical agreement, or CA. So this is really the number of isolates where the susceptibility testing system that you're evaluating gives the same category interpretation as the reference. And by category interpretation, I mean susceptible, intermediate, or resistant. And you divide that number of isolates that had the same category result by the total number of isolates. And ideally, you want your categorical agreement to be greater than or equal to 90%. Essential agreement is essentially getting at the number of isolates where the susceptibility testing system that you're evaluating has an MIC that falls within the same MIC or one doubling dilution on either side of the MIC of the reference MIC divided by the total number of tested isolates. And again, you want that greater than or equal to 90%. Some of you might be thinking, oh, that means that we accept a 10% error rate in our susceptibility testing systems, and that is absolutely true. We then talk about different types of errors for susceptibility testing systems. So the first is a very major error, and I like to think of this as false susceptibility. So that's where your susceptibility testing system that you're evaluating called an isolate susceptible, but in truth, it's resistant by the reference method, which would be very bad, which is why we call it a very major error for patients. And we want that error rate to be less than 3% by CLSI standards or less than 1.5% by FDA guidance. Major errors, on the other hand, are false resistance. So that's where the test system that you're evaluating calls your isolate susceptible resistant when in truth the reference method calls it susceptible. And you want that major error rate to be less than 3%. And then finally, minor errors are where you have one method that gives S or R and the other method gives intermediate. And we call those minor because it's not as bad to be going between susceptible to intermediate or intermediate to susceptible as it is for like a very major error where you're going from resistant to susceptible. And we want those minor errors to be less than or equal to 10%. So like I'd mentioned, the phenotypic susceptibility testing method that we evaluated was the Phoenix M50, and we compared that to the broth microdilution result from Public Health Ontario. The Phoenix M50 was evaluated with their NMI C451 panel, and this panel is actually already in use at shared hospital laboratories for routine Pseudomonas susceptibility testing of first-line agents like ciprofloxacin, piptazo, ceftazidime. There is a ceftolozane tazobactam and ceftazidime avibactam that are on the panels, but those have not yet been validated. So that was the purpose of this work. And importantly, there's no published literature on clinical performance of this panel for susceptibility testing of these agents. So this was the first clinical evaluation of those susceptibility testing method for these agents. And then unfortunately, imipenem was not on the panel, so we could not evaluate it with the system. We tested all 183 isolates on the Phoenix, One isolate repeatedly failed to grow after 3 attempts on the Phoenix panel. So overall, we had 182 isolates where we had a paired broth microdilution and Phoenix result. So here I'm showing the results of the evaluation after discrepancy resolution. So whenever the Phoenix and broth microdilution give discordant results, it's important to try to resolve that discrepancy. Ideally, both methods would be repeated, and both would be repeated with the same inoculum. Unfortunately, it was not possible to repeat broth microdilution at Public Health Ontario, so discrepancy resolution was only done by repeating the Phoenix 3 times. So I think it's important here to acknowledge that in some cases, the Phoenix might have been right, and the error might have laid with the gold standard broth microdilution, because we also know that there's some variability in broth microdilution testing. So really, that broth microdilution testing would ideally have been repeated as well for discrepancy resolution. We'll first start with ceftazidime avibactam. So the essential agreement met acceptance criteria. It was 90.1%. Categorical agreement, on the other hand, was unacceptably low at 83%, and that was mainly related to a high number of very major errors. So the Phoenix called the isolate susceptible, but public health called the isolate resistant. Many of these very major errors were within essential agreement, which is reflected in the acceptable essential agreement rate. Ceftolozane tazobactam had an unacceptably low essential agreement and categorical agreement as well. Most of this error was in minor errors, although we did have 2 very major errors. There were no major errors with ceftolozane tazobactam. I think that there's 2 things to highlight here that can cause problems whenever evaluating susceptibility testing methods. So the first is, again, that we were unable to repeat broth microdilution, which can be the reality for many clinical labs that are trying to evaluate these commercial systems. So that does pose problems for academic or hospital-based laboratories that are trying to evaluate these systems, because broth microdilution is not routinely performed in our hospital-based laboratories. The next that I want you to note is that there is no intermediate category for ceftazidime avibactam, which is why there's no minor error that could be calculated. If there were an intermediate category for ceftazidime avibactam, many of these very major errors could have been minor errors. So we do run into issues of very high, very major error rates or major error rates whenever we're testing an antibiotic that doesn't have an intermediate category as a buffer to those very major errors or major errors. And then finally, I just want to say that all of these acceptance criteria assume that less than 20% of isolates are around the breakpoint, which was clearly not the case here. It's very easy to evaluate susceptibility testing methods whenever you're evaluating a panel of isolates where the MIC is really low, and then some of them have an MIC that's really high, and you don't have very many isolates around the breakpoint. When you start having very many isolates around the breakpoint, that's when you start getting into issues of these high error rates, even though the essential agreement is not too bad. So here I'm just showing you those MIC distributions again, just to highlight for you that quite a significant proportion of the ceftazidime avibactam isolates that we tested have an MIC around the breakpoint for susceptible and resistant. And the same is true also for ceftolozane tazobactam, where you can see quite a number of isolates around the breakpoint for the intermediate category, which was reflected in the high minor error rate that we saw. Because of these challenges, there is a method that has been developed to validate susceptibility testing methods when there are more than 20% of isolates that fall around breakpoints. And this is a linear regression model called error rate bound method that accounts for the expected variations in MIC testing by allowing for a higher error rate around the breakpoint. So you're basically just changing your acceptability threshold criteria depending upon what the MIC of the isolate is. So here I'm showing ceftazidime avibactam first, where if you look in this middle row here of all of the isolates that are around the breakpoint, you still have a very high, very major error rate. So with the error rate bound method, you would accept a 10% very major error rate in this MIC range, whereas we still see a very high, very major error rate. The major error rate becomes acceptable with this error rate bound method because we accept a 10% major error rate in this MIC range. And again, you can't calculate minor errors because there's no intermediate category for ceftazidime avibactam. And then for ceftolozane tazobactam, suddenly the performance of the system from an error standpoint becomes acceptable. So we see that our very major errors were all within twofold dilution of the breakpoint. And we see that it's less than 10%. It's only a 2.5% very major error rate there. And then for minor errors, we accept 40% minor errors in this MIC range around the breakpoint. So the 29.6 minor error is less than the 40% that we would find acceptable by the error rate bound method. So overall, ceftolozane tazobactam performance probably acceptable by the Phoenix, given the very challenging isolate panel that we used for the validation. Whereas even with giving the ceftazidime avibactam susceptibility testing a bit of grace with this error rate bound method, ceftazidime avibactam resistance is still severely undercalled by the Phoenix. And really, this means that we need more rigorous paired analysis where you are directly comparing Phoenix with broth microdilution using the same inoculum on the same day. The next thing that I wanted to talk about was our evaluation of genotypic susceptibility testing for all three of the beta-lactam beta-lactamase inhibitor agents. So we did whole genome sequencing on all 183 isolates that we evaluated on the Illumina mini-seq, sorry. And the resistance genes and mutations were identified with the NCBI AMR Finder Plus tool. There is a gigantic disclaimer on the AMR Finder Plus that says for research use only, not for use in clinical diagnostic purposes, which I totally acknowledge. And this was purely for exploratory analysis. And do not worry, this is not being used for testing of clinical isolates. We defined genotypically resistant isolates as having a known determinant that confers resistance to ceftazidime avibactam, ceftolozane tazobactam, and or imipenem that was identified by AMR Finder Plus. And then isolates that were genotypically susceptible, we defined as having no known determinant that was identified by AMR Finder Plus. Just a short pause to discuss the resistance mechanisms to these three agents. So the first thing to highlight is that while ceftolozane tazobactam and ceftazidime avibactam are supposed to be active in the setting of PDC upregulation, there are some PDC variants where upregulation of these PDC variants can still confer ceftolozane tazobactam or ceftazidime avibactam increased MICs, although whether those increased MICs translate to clinical resistance is still a topic of discussion. MexAB OPRM expression can confer ceftazidime avibactam and imipenem. The mechanism for MexAB OPRM overexpression for ceftaz-avi is efflux of ceftazidime. And then for MexAB OPRM with imipenem, it's actually efflux of the relebactam. The other important resistance mechanisms to highlight here are these AMP-C omega loop mutations. So these are mutations in the omega loop that basically gates access to the catalytic center of the PDC. And these mutations are known to confer resistance to CT and CZA. And then finally, I just wanted to highlight the carbapenemases, metallobeta-lactamases, like VIM that Megan was talking about, or like NDM, confer resistance to all of these agents. And then the class A carbapenemases like KPC confer resistance to ceftolozane tazobactam because tazobactam is not a good inhibitor of KPCs, whereas avibactam and relebactam are. The only thing that this table is missing is that there are also PBP3 mutations that confer resistance to all three of these agents, and PBP3 is the penicillin binding protein that is bound by all three of these beta-lactams and is the essential penicillin binding protein in Pseudomonas aeruginosa. Just to quickly highlight the mechanisms of resistance that we saw in our isolate that were uncovered by AMR Finder Plus. So only 3 isolates reassuringly had carbapenemases. So it still remains true that at least in our setting, carbapenemases are a rare cause of resistance in Pseudomonas. We did see a KPC2, we did see an NDM1, and we did see a VIM5, and all of those, their susceptibility testing results by broth microdilution made sense with the enzyme that was identified. There were a couple other acquired beta-lactamases, which are truly extended spectrum beta-lactamases with ceftolozane activity that were identified. Surprisingly, we saw no PDC omega loop mutations, which was surprising because in many jurisdictions, these are actually a really important source of resistance after carbapenemases. We did see a number of mutations that were predicted to upregulate MexAB and PDCs, but we didn't perform any expression studies. And then we also did identify PBP3 mutations that are known to confer beta-lactam resistance. Taking all of our genotypic predictions based upon the AMR Finder Plus output with what we were seeing phenotypically by broth microdilution, we see that the categorical agreement for ceftolozane and imipenem were not too bad, but that's obviously at the expense of there were still quite a number of very major errors for all agents. And I think that really just reflects that not all resistance mechanisms for these agents are either known or curated in the AMR Finder Plus database. Importantly, whenever we did this analysis, you'll note that I excluded intermediate isolates by broth microdilution because we did not define an intermediate genotypic susceptibility testing category. Some people will combine intermediate with resistance for this type of analysis, but that's not really the purpose of the intermediate category, so we did not do that for this study. In terms of moving forward with next steps for genotypic susceptibility testing, it's good that acquired beta-lactamases remain rare in our setting. Genotypic susceptibility testing does perform much better in settings where carbapenemases are very common, and most of that work has been done in Spain. There's still a lot to learn about mechanisms of ceftazidime avibactam, ceftolozane tazobactam, and imipenem resistance. It's a very complex multi-genotypic phenotype that's not fully understood or curated. Interestingly, we have started doing a little bit of a secondary analysis of our genomes to see if we can start identifying new resistance determinants that have not previously been identified. So we did identify 2 new PDC variants that were identified in resistant isolates. Whether these have a role in phenotypic resistance in these isolates requires experimental validation. So if you have an honors student or a master's student who would like to test that hypothesis, please let me know. I'd be happy to collaborate. And then finally, given that a lot of the resistance that we're likely seeing is due to expression-related changes in MexAB, OPRM, or PDC, probably a multi-omics approach will be required to supplement genotypic susceptibility testing in Pseudomonas aeruginosa in particular before this sort of genotypic susceptibility testing is able to replace phenotypic susceptibility testing. And with that, I'd just like to acknowledge the team at Sunnybrook that I was working with, in particular Zena Lee and Rob Kozak, who are microbiologists at Shared Hospital Laboratory. And then all of the people in their lab who were very helpful for doing the sequencing and the susceptibility testing. And for some of you, Nick and Finley might be familiar. And with that, I'd like to say thank you.
Thanks very much, Clayton. Really super interesting talk. So we have a question from Jake Kaladi for Dr. Hall. Excuse me. Were all of the PBP3 mutations you identified in the transpeptidase domain, or were there others in more distal regions like the pedestal or transmembrane helix?
All of them were in the transpeptidase domain.
Straight up question, straight up answer. Okay, I don't see any more questions there right now, so I have a few of my own. For Megan, did you guys look at the effect of adding zinc chelators? So, in other words, could you further sensitize the carbapenemase producers by adding or removing zinc even further?
Yeah, we did. We did a lot of in vitro work, showing that zinc chelators also chelate. I think that's also the same mechanism that AMA, discovered by the Wright lab, uses.
Right. Yeah.
We didn't do any in vivo work, though, knowing if you paired like azithromycin with a zinc chelator, if that became even more potent.
Hmm. That's very, very interesting. So I also have a question for Clayton. I was struck by how long it takes to get microbroth dilution testing results, and the Phoenix clearly, excuse me, is faster. But is one run on the Phoenix enough, or would you? So basically, how much faster is it, and is a single run enough to give you confidence that you have the right answer given the results of your study?
It's a great question. Public Health Ontario, the turnaround times are around 7 to 10 days. It's not that the broth microdilution takes that long to perform. Broth microdilution takes, an overnight incubation. It's more so like the processing and the shipment and
Hmm.
the actual reporting of results that's taking the time. The Phoenix system can give a result within 24 hours as well. The convenience for it would be that we have it on site, whereas we don't have
Hmm.
broth micronization on site. Given the performance issues that we had, it's not being used clinically for that purpose now. So we are investigating other phenotypic susceptibility testing methods apart from the Phoenix. It would have been convenient if the Phoenix would have been more reliable because it's being used routinely for normal susceptibility testing of Pseudomonas. So you would have gotten the ceftazidime and the ceftolozane tazobactam at
Hmm.
the same time you would have gotten the piptazo. But unfortunately, it didn't work out that way.
Hmm.
Whenever manufacturers of susceptibility testing methods are verifying their method, they're typically not using isolate panels that are really challenging. So they're
Hmm.
using isolate panels where a lot of them are really susceptible, a lot of them are really resistant, but you're not really having these isolates that are in between. And we chose to use a more challenging isolate panel because this reflects the actual isolates that we want susceptibility testing on. It's not really of
Hmm.
value to test a bunch of susceptible isolates that would never be treated with these agents anyway.
Hmm.
So unfortunately, that's where we ran into problems with the validation was we were probably just taxing the system a little bit too much within the realm of what would be expected variation of susceptibility testing.
Yeah, I was just struck by, it takes long enough to get these new drugs through special access, and then if you pile on the length of susceptibility testing, and you probably can't necessarily do those things concurrently. Okay, we have time for one more question from Merrick. He says, rather than setting a gold standard, could the Phoenix public health and genotypic resistance predictions be analyzed using latent class analysis? Ooh, heavy
That's
question.
a great question there. Happy to chat offline about it. I actually do not know what latent class analysis is, so that's an interesting question. I assume that it's really getting at can a gold standard be a combination of methods? And for some antibiotic agents, that's probably the right way to go. And a good example of that is piperacillin and tazobactam, where really I struggle to say that there's any good gold standard for susceptibility testing. We know that there's tremendous variation in broth microdilution testing, particularly for piptazo. So it's a good question and is probably something we should think about a little bit more.
Okay, we are at the end of our hour, so thank you both very much for those fantastic talks. Our next rounds is not until December, and I will see you then.